And the speaker will be Simona Di Gregorio of the University of Pisa. She's an environmental microbiologist in the Department of Biology, and she coordinates a group of young scientists, PhDs and postdocs doing research on bio-based processes dedicated to the decontamination of environmental matrices. So the main focus of her research are the contamination of soil and sediment contaminated by, for example, hydrocarbons or pesticides. The title of the presentation is Bio-based remediation, hydrocarburoclastic fungi and bacteria to improve bioavailability and degradability of petroleum hydrocarbons in an historically contaminated soil. Please, Simona.
Thank you for your introduction, and I must thank you for inviting me to join this meeting, very interesting meeting. And now I start to annoying you with subjects that is not the core of your research. But anyway, perhaps we can find some traits, for some ideas for a sort of collaboration between the two fields. Okay. So, yes, what I'm going to tell you this morning is something about the teamwork among fungi and bacteria in soils that are contaminated by total petroleum hydrocarbons. And I realized with Professor Lopez that the concept of the microbiome that is doing something for the environment is already consolidated in your group, while when we talk about high level of contamination by hydrocarbons or pesticides, but especially by hydrocarbons, the people that are dedicated to the bioremediation of the environment are more focused on what we call the specialists of degradation. So we try to isolate microorganisms that are capable to use that source of contamination as a sole carbon sources, and then we do bioaugmentation. We increase the amount of that microorganism in the environment to see a degradation of the contaminants. But actually, the contamination of the environment is always a teamwork. It's not, the merit of only one microorganism or the other. And of course, I think also in the case of the accidental dispersion of plastics or biopolymers or bioplastics in the environment, of course you have to consider which are the microorganisms that are facing that situation. And especially with disturbed matrices, disturbed environments, what we see in relation to fungi is that they are more colonized by Ascomycota. You are not microbiologists, but this is quite strange because when you talk about bioremediation, usually you see the exploitation of Basidiomycota, that are the edible mushroom actually, because they degrade lignin, and the lignin is really very recalcitrant to biodegradation, and they produce a lot of extracellular enzymes with high redox potential that can be exploited for the degradation of aromatic fractions of hydrocarbons, but we can think also about the aromatic fraction of plastics or biopolymers or whatever. While the environmental matrices that are disturbed are mainly, the dominating phylum of fungi are the Ascomycota. And the Ascomycota are very important in the environment because, they are not very studied in relation to the remediation processes, even though they have been described as capable to transform also recalcitrant. And they are very important in the environment because of their saprophytic metabolism. So they are responsible for the synthesis of organic matter in the soil and also for the smooth release of carbon sources deriving from the recalcitrance of the humic fraction. So they are producing enzymes that are capable to depolymerize what is recalcitrant to biodegradation and might be also eventually biopolymers or plastics, petroplastics are the main problem in the environment, that might show some chemical moieties that are similar to the humic fraction. But they also produce enzymes that smoothly depolymerize and so produce radicals. Radicals that tend to stabilize by polycondensation reaction. As chemists, you know very well this kind of reaction, I think. And these are the base of the synthesis of humic fraction, so the humic acid fraction. So they are doing both aspects. They are mobilizing and stabilizing the carbon sources in the environment. Usually we face problems like contamination by total petroleum hydrocarbon. In this specific case, we were working in a decommissioned oil refinery sites in the north of east of Italy, and we were facing a contamination of 8.5 ppm of total petroleum hydrocarbon. That is a medium level of contamination, but it's critical, of course, for the environment. Limit says something like 10 ppm for civil environment and industrial environment 100 ppm. So we have really to cope with a very high level of contamination. So what we wanted to do, we wanted to design a process based on the exploitation of the metabolism of the Ascomycota. There are actually the dominant fungal species that is colonizing this kind of environment. And we wanted to exploit either the carbon mobilizing activity and the carbon stabilizing activity of these biological moieties. And then, of course, we want to study the synergies between fungi and bacteria. Okay, so we isolated, as I've seen from Professor Lopez, so we isolated a series of Ascomycota, and one of these was quite interesting because it was a Ciboria species that it's not really characterized in the environment. It's very rare in the environment. So for this reason, for a biologist, of course, it's of a certain level of interest. And then it was capable to use diesel oil that we can consider a sort of model of TPH contamination. It can use diesel oils as sole carbon sources. So, sorry. I always do the mistake with the pointer. So it can use diesel oils as sole carbon sources and produce a quite reasonable amount of biomass to do eventually to design a bioaugmentation approach. And it has been previously characterized also in wastewater treatment plants as capable to transform polymeric azodyes and aryl amines. And in our case, since we are facing with the aromatic fraction that is more recalcitrant to degradation than the saturated fraction of TPH that might be of a higher interest if we talk about biopolymers. I mean, this fungi was capable to produce either manganese peroxidases and laccases, so polyphenol oxidases, extracellular polyphenol oxidases that can be exploited in our processes. So it was quite interesting. And so we design, we do bioaugmentation. I think we can do bioaugmentation of competent microorganisms also in soil when we use biopolymers for mulching or when we compost plastics or bioplastics. So we do bioaugmentation, and the sets of experimentation was 3 mesocosms only biostimulated, and adjusting carbon, nitrogen, and phosphorus sources, the ratio for biodegradation. And then we augmented the Ciboria species at 1% fresh weight ratio or 7% fresh weight ratio. Well, we measured TPH content, that means TPH depletion eventually. And then we measured also the stabilized carbon form, that is humic and fulvic acids content, because we wanted to see if the fungi was capable to degrade or to stabilize the contamination, so humification or degradation. And what we did usually is a metabarcoding, so we want to know who is there. Either has a bacterial moieties and fungal moieties. And we do, in soil, we do productive functional metagenomics to see who is there is doing, especially for the bacterial moieties. As Luca yesterday showed you with the degradation of PBS. Okay, so some results quickly. If we adjust only the ratio on carbon, nitrogen, and phosphorus, we do not have any depletion of TPH in 90 days of incubation. This is not favorable to design a bio-based process that has to be transferred on industrial scale, while we have depletion of TPH in 90 days with 1% of amendment by augmentation of Ciboria. The kinetics is more favorable in the case of 7%. Okay, and in the case of 7%, we have also a significant increase in the humic fraction, so in the polymerization of the organic matter. Okay, so this means that the fungi microaugmentation is mandatory for the depletion of TPH, because we do not see any depletion, at least in time that can be transferred on an industrial scale, of course, in nature it might occur, but in very long times. Okay, I'm not going to tell you much on this heat map. They are very nice, but very difficult to understand what they mean. They mean that the Ciboria was present at the end of the process, so it was mandatory, and it was there at the end where we observed TPH depletion. I show you again this dendrogram. That means that we have sort of segregation of the ecology of the bacterial community. So we have a bacterial community that is different between the Mesocosms at the beginning of experimentation. At the end of the experimentation, we see a different segregation. That means that we have different bacterial populations that are involved in these two stages. And then we have a sort of segregation of what we define the transitional Mesocosms. The Mesocosms where something was occurring that was depletion with a certain kinetics, with a more favorable kinetics, or not TPH depletion at all. And they are all together. That means that the bacterial ecology in some way is related to what we observed on a chemical point of view. In fact, if we look at the fungal ecology, I will go fast, we can read only the sentences. Any significant changes in the fungal ecology during the process of TPH depletion. This is contradictory because we have seen that the fungi augmentation is mandatory for, but there's not any significant changes in the ecology. That means that, in a way, someone else should help these fungi to do the job, because they do not change in a significant way, and they are not correlated to the depletion of TPH, or slightly correlated to the accumulation of humic and fulvic acids. So there seems to be much more involved in the humification processes, but this cannot be so important for the depletion of the TPH. If we look at the bacteria, we have on the other side a significant changes in their ecology. So that means that they change during the process of TPH, and so perhaps they are participating to the process. And in fact, they have a significant correlation, the ecology has a significant correlation with both TPH depletion and the accumulation of humic and fulvic acids. So there's a sort of teamwork, as I said at the beginning, and so it would be important to have in our hands not only the fungi, but also the bacteria that are responsible for. To see what they do, we do predictive functional metagenomics. We could do also genomics. I don't go in details, but in soil genomics, metagenomics is really difficult, so we have a predictive approach. But in other processes dedicated composting, or I don't know, other processes dedicated, for example, to the treatment of biopolish, it might be possible to do also genomics and not only predictive metagenomics. So what we see with these instruments, we see what the bacteria that are in the matrices, what they can do. They can do. And you see these names are names of recalcitrant molecules that but the bacteria can degrade. And they also respond in different, I mean, if we consider the xenobiotic biodegradation and metabolism module, so all the enzymes that might be involved in the degradation of TPH, they are changing in the different sets of mesocosms. They are responding, I mean, they are responsive to the treatment. So bacteria are definitely involved. And briefly, what we see, that's why I was asking to Professor Lopez who they are, because for a microbiologist, who is there and what is doing is important. And we see, since with the metabarcoding we have the name of the genus of bacteria that are inside our mesocosms, we see the blooming of interesting bacteria that are actually, I go a little bit further. Yeah, sorry. That are actually expressing one interesting enzyme that is a peroxidase, a bacterial peroxidase with a quite redox potential that is expressed, and its expression in a way is mandatory for priming the TPH depletion. And also the blooming of what I mentioned at the beginning of this talk, the specialists, so bacteria that are described to use the TPH as a sole carbon source. So we have bacteria that are expressing, actinobacteria mainly, that are expressing these enzymes that are mandatory for TPH depletion and for the blooming of the ones that are described as responsible for the degradation of the TPH. Okay? But if we look at the statistical analysis, we do not have any correlation between these enzyme activities that seems to be interesting and the results that we obtain in relation to TPH depletion. So the enzymes is not directly involved in TPH depletion. It is actually involved, because it is a peroxidase, in the saprophytic metabolism of the actinobacteria. So we have fungi and bacteria that are not focused on the degradation or the contamination, but our hypothesis is that they are involved in the mobilization of the carbon sources, that in an aged contamination, because this was a decommissioned oil refinery, might be involved in this evidence that when the contamination is aged, it is not bioavailable. If something is not bioavailable, you cannot degrade. So these microorganisms are much more involved in the improving increment of the bioavailability of the contamination, and they are so mandatory for the blooming of the specialist, because if they provide the carbon sources for the ones that are really involved in the degradation of the contamination. Okay, so nothing to say to people that are involved in the biopolymers. But I think that's a very important concept, at least for us, but I think that we can also, I mean, the spillover might be found also in your sector, or what should be done to close the cycles of the production and degradation of biopolymers in the environment, is that, of course, we have to exploit the teamwork between different functionality in terms of microbiology. And in our case, it was mind the generalists, because really we are focused on the isolation of specialists, and we don't think about generalists that are responsible for improving, for example, the bioavailability of the contamination. And actually, well, this is the product of several years of work, and it ended up with the, well, we gained an innovation action that started next October, and it will end in 227. And it is, it is based on the application of artificial intelligence to integrate the genomic data, metagenomic data, to see who is there, what is doing, with the control of chemical, physical parameters that are involved in the process dedicated to the decontamination of soils and sediments. We work in teamwork with companies that are involved in this kind of processes at the industrial scale. And I'm coming back from the bioremediation conference, where bioplastics were one of the subjects. And I would say, this was one of the slides that have been shown by many of colleagues. There are points that are very clear to your group of researchers. But what I want to say is that, as far as I understand, the end of life of bioplastic is quite important, and it's quite important to design processes that are not only biodegradation, because the biodegradation means actually the depolymerization. That might means also the production of microplastics, nanoplastics, and something that has not been really mineralized. While composting processes, I think they are very, very important. And so the reinforcement of the composting processes also at industrial scale, or in the composting plants that you can have in your backyard, in your home, when we are composting the organic fraction. But anyway, it's really important to consider this aspect. And I hope that the future goal of biopolymers would be the toolbox for bioplastic remediation, and not only for soil remediation and sediments. So, thank you for your attention. I think I annoyed you a little bit with arguments that are not really your core business, but I hope.
Thank you, Simona, for your nice presentation that is also suggesting to have a multidisciplinary approach to these topics.
Yeah, I think it's important. It's main focus.
So I ask if there are some questions.
Hello, my name is Francisco Villaplana. Thank you very much for the very interesting presentation. I really enjoyed it. I have actually a couple of comments. Now that you have this slide here, when I'm teaching in Sweden about biodegradation, we do not only talk about the polymerization, but also about assimilation of the degradation products by the organisms.
Yeah.
How does that fit with the definitions that actually are shown in the standardization organizations? Does assimilation also is included?
Yeah, do you mean in relation to biopolymers or to?
Yes, in biopolymers.
Well, I assume yes. But yeah, it is or not in these slides. Yes, yeah. You're right. Yeah, well, it's a very interesting question, but perhaps I do not have the proper answer because I'm not involved exactly at the moment. I hope it will be in the future. Yes, assimilation. Yes, assimilation, of course, but if you assimilate, you transform something that is in something that is biomass and should be neutral at least to the environment. I don't know if this is true because for petroplastics, I don't think this is really true, or it should be studied at least, or should be studied, as we said yesterday, we should be studied which is the impact of this assimilation on the microbiome of the organisms. And that means also that microbiome is involved in interaction between the organism and the environment. And if the microbiome is deeply or in some way changed, we should evaluate which are the effects on the biodiversity, first of all, and all this spillover of decrease or changes in biodiversity in the environment. So, I mean, I think your question is really relevant, and unfortunately, we do not have still the answer. We don't know.
Yeah, and I agree with you. I think this is a very fundamental topic of research. But anyway, we can talk later because it's a long. My second question, the one that I originally had, was you showed a lot of oxidative mechanisms in your in the bioremediation studies, and I think that's going to be critical as well for polymers. So do you have any thoughts, or do you think that the same organisms and enzymes that are involved in your process
definitely.
could be?
Yes, and in a way, Luca was showing yesterday these particular traits. I mean, when you talk about TPH, actually you talk about a mixture of contaminants that are saturated and unsaturated, and the enzymes that are involved are hydrolases or polyphenol oxidases or lactases. So, of course, the same bacterial enzymes are involved in the degradation of biopolymers and also petroplastics. Actually, petroplastics are deriving from petroleum. So, if you have microorganisms that are coping with petroleum, you should have the same microorganisms that can cope also with petroplastics. Unfortunately, the external parameters are not optimized for a process that can be transferred on industrial scale, perhaps. And so the recalcitrant is a matter, because then it has an effects on the biodiversity, on the environments, and long effects, and that's the main problem of the leakages of these recalcitrants in the environment, uncontrolled leakages of these recalcitrants in the environment. But of course, I was really fascinated by the fact that Professor Lopez was transferring bacteria from the gut that is in an aerobic environment to the aerobic environment. So you see bacteria and microorganisms at the end, they can degrade everything. And one of the main points, for example, of the bioremediation conference was the capacity to degrade antibiotics. So, I mean, microorganisms are really our best friends in a way, but they can also be very dangerous, especially when they degrade and do not mineralize, because if they do not mineralize, they release toxic components in the environment. So yes, anyway, the microorganisms are the same. So the same bacterial enzymes can be exploited also in these processes. Okay, thank you. There's a question there.
Thank you for that. Thank you for that lovely talk. Sorry, I missed the beginning, but I was thinking at an industrial scale, with your knowledge base now, can one develop a microbial kind of consortium to start degrading in a very controlled manner the various polymers that we have? Is that something that's happening?
I mean, in relation to these innovation action, that's the final goal. We have a lot of microbial candidates, and we want to isolate macrobiota, and not only microbial candidates as sole candidates. And yes, actually, we design these processes, and we start from lab scale, and we assess the processes at least at the pilot scale. So there are the instruments for. The point is to integrate the physical parameters of the processes on a real scale to the metabolism of this macrobiota. That's the main point. And in soils and sediment, of course, this is quite we are quite far, still quite far, because the processes are not so consolidated, actually. But I think that in other kind of processes that you are studying, this might be much more possible, because the processes are more consolidated, like the composting of the organic fraction or whatever. While the co-composting of bulking agents in soil, it is something that is applied, but is not so consolidated in the regulation of the chemical and physical parameters. That's why we would like to reach this goal, and we hope we will in these 4 years, 5 years of research. But it's the main point, because you can have your microorganisms, but if you don't transfer their metabolism on the real scale, they won't help you, of course.
So almost like you could make them like the plastics by, almost a metabolic adjustment to what's around, and then use that.
Yes, actually, in this innovation action, there's a company that is doing this piece of work. They do a sort of artificial selection, speed up of the selection of the microbiota metabolism. There's very much. Altar is the name of the company. They're very.
What's the company, sorry?
Altar.
Altar.
I can give you some information, because might be of interest also in other sector.
Yes, absolutely. Thank you. Thanks.
Welcome.
Very interesting. I have a lot of questions, so maybe I will try to catch you later on. But referring to this last slide here, I'm not quite sure that I agree or I understand what is the difference between biodegradation and and composting in the sense for polymers. Because when you have molecules like hydrocarbons, low molecular weight materials, degradation attacks the whole thing, let's say. Okay, so the whole molecular structure in some way, in different ways, of course. Polymers is different. Polymers is a solid particle, so it has a surface, may or may not have porosity, and degradation may attack. So biodegradation is not necessarily depolymerization; can be fragmentation. And then that has, involves fragmentation of the solid particle into smaller fragments, and then exposure of surface.
Yes.
And then surface is exposed to biological attacks and so on and so forth. So it's a more complex
No, yes, you're right. But, I mean, try to understand my point of view. I'm not an expert in the sector. But what I see is that the problem with plastics is, at least with petroplastics, are micro and nanoplastics. It is true that microplastics are more exposed to the action of the microorganism, but then regulating these processes in the environment is not so easy. That is the same that we have with Total Petroleum Hydrocarbon. What I show you is not a mineralization of TPH, it's a polymerization, humification of the fraction. That is not mineralization. And humification means that you have to do a lot of toxicological assessment, because what I have in my hands is that a humic fraction that derives from the contamination. I mean, for people that are involved in this kind of processes, it can be, well, they say, okay, we reached the goal, because the goal is the limit of the law. But then we have a lot of problems. With polymers, I think it's my opinion, we have the same problem. I mean, we have degradation that is depolymerization. If it's not mineralization, the product is not humic fraction that had to be evaluated on a toxicological point of view. But I would say that it's easier because you do the test, you do dedicated tests during time, with time, like dump sites. I mean, or landfill. When you close a landfill plant, then you follow the toxicology of the environment for 30 years. And you can do the same when you are in a dismissed refinery. You can follow the destiny of what you have done for tens of years. But with biopolymers, at least with plastics, if you generate micro and nanoplastics, you cannot really control the process of dispersion of this stuff in the environment. You know that you expose these small molecules to the surface, the ratio between the surface and the environment is in favor of mineralization. But we do not have any instruments to follow it. So we should think about these aspects.
So if I can, just from my understanding, which is limited, of course, in this area, the direction towards complete mineralization, as opposed to humification, is microorganism dependent, is substrate dependent, is both. So what is it?
Both. It is both. And so it might be also with biopolymers, perhaps. Not with bioplastics, with petroplastics, but with biopolymers, yes. Yeah. If I understood your questions, I'm not, but you're right.
Thank you, Simona.
Beatrice, I'm sorry. Excuse me. Excuse me. Excuse me. Simona, if you can get back, there is Professor Maria José López from the University of Almeria. So you got plenty of questions.
Sorry.
Yeah.
Sorry about that.
Yeah, I have so many questions and so many things to discuss that I will ask Patricia next year if he's organizing again this conference to add a session for the microbial part, because, well, thank you very much, Simona, for your your fantastic presentation. I'm very interested. And I think that you are a bit late with the schedule. So I'm plenty of questions and things to discuss with you. Congratulations, because the things that you are doing are very difficult to perform. I guess that the first experiments were performed in a containers using this inoculant that you selected, that in fact we are also using as Ascomycota fungi instead of Bacillomycota,
Yes.
and this is some common point. I think we need to discuss and to
Happy.
to interact, because I'm thank you very much. I don't want to go very much through this one, and also in the bioplastic and the plastic metabolism, and also in the consortia, which I think that is the future for this, and the
Yes.
analysis of the ecosystem, which is very complicated. Yes, I wanted to point out this. Thank you very much, Simona.
Thank you.
Thank you.
Thank you, Professor Lopez. I hope to see you in presence and to discuss in presence.
That's for sure. Thank you.